one but two Storer lecturers. Today's lecture is, of course, Dr. Caroline Dean, and tomorrow I want to make the point that there is a second Storer lecture at noon in the life sciences building. That's Jonathan Jones. On some topic I can't pronounce the name, but you know Jonathan Jones by the way, so it's on fungal diseases. That's at noon tomorrow. So the Storer lectures thankfully allow us to bring people from far-flung places of the British Empire to and so today we have Caroline Dean, who's from the John Innes Institute. Caroline got her PhD in the University of York, and then came to California to do a postdoc with John Bedrock, who's then with a company called AGS, or Advanced Scientific Systems. It then became DNAP. Then been bought over, but had some of the great, had a fantastic set of plant biologists working at the same time. Hugo Lunar, Tal Malaga, other people, really great plant biologists. And so Caroline worked with John Bedrock's group on small subunit genes in plants, which has not much to do with what she did in her faculty position now. At that time, that's when she tried to grow tulips and found out that they needed a period of cold treatment and began to wonder why plants need cold to flower. And when she got her position, she's a long-standing collaborator and colleague of mine. And at the time she went to the John Innes, I asked her what she intended to do. She said she wants to figure out what makes the requirement of cold for flowering in some plants. And that was 2 decades ago. She stuck to that. Unlike many of us who've gone from problem to problem, she stuck with essentially the same problem and the same 2 or 3 genes and learned wonderful things in all this time. She'll tell you about. And in her seminar today, which is on visualization and chromatin silencing, as you all know from our papers and publications, she's done some of the seminal work in this area. She's been at the John Innes Institute during all this time, since she left California. She informs me that she's no longer associate research director. She's out on parole for administrative duties for good behavior. But she's still at the John Innes, and she's been a project leader still, and she's received numerous honors such as the Embo Fellow of the Royal Society, the foreign member of the U.S. National Academy of Sciences, and this is most impressive. And she's an officer of the Order of the British Empire. And on behalf of the former colonies,
well, it's lovely to come back and see everyone. I have a lot of old friends here, so it really is a wonderful, wonderful invitation. Thank you. And always very nerve-wracking to be introduced by Sundar. So, I left my gown at home and I trekked from the far reaches, but I actually made it here. So, anyway, so what I'm going to do today is tell you our work on this process that, yes, I have been studying for a very long time, on understanding the timing of flowering in plants. And it's an example of a developmental transition that, like many others in plants, relies on monitoring and seasonal cues to get it right. Because timing is really important, as the slide says. If plants get the timing, their developmental timing wrong, the timing of flowering wrong, then there's fairly disastrous consequences for reproductive success. So many pathways have evolved for allowing plants to monitor exactly the right time to flower. There are environmental cues monitored, and there are endogenous cues. And, for a long time, people thought, gosh, when different plant species, many of you may be gardeners, some plant species need particular photoperiods, other need temperatures. There are lots of different cues. So while there are a lot of different mechanisms, And so for a long time, people really didn't understand much about the molecular controls of flowering. But what's happened over the last 15 years or so has been a concerted approach to try and take genetics and molecular analysis to understand flowering in Arabidopsis, the reference, and then to use that information and understand, flowering on a broader context. So the next slide really is the simplest version of flowering time control that I can draw for you. And it's just to remind me to say that, there are multiple inputs. Some of those inputs are seasonal cues, some are endogenous cues, as I said. Some promote flowering, some repress flowering. But in essence, they all focus in on regulating a common set of genes that have been called floral pathway integrators. And it's the quantitative expression of those that determines when the plant switches to flowering. And these very different physiological requirements of different plant species are really because of different quantitative inputs of these different pathways. Now, the one pathway I'm going to describe today, and the two that we've been studying mostly, are these two at the bottom here. So I'll tell you about how winter affects flowering. Winter represses a floral repressor, so if you repress a repressor, you're going to promote flowering. And that's what I'll talk mostly about today. I'll very briefly mention this one here, which is just randomly called development, but I'll explain it later. So this is the process of vernalization. It's a quantitative process, so increasing weeks of cold, you progressively accelerate flowering. Shown for Arabidopsis here, this is a late flowering genotype. With no cold, flowers very late. With increasing weeks of cold, you see this progressive acceleration of flowering. It's reversible, it's perceived by dividing cells, it's not graft transmissible, unlike the photoperiodic signal. And these properties at the bottom here, this mitotic stability and this resetting at meiosis, many years ago suggested that vernalization may have this epigenetic basis. Now, back in '99, I should say, of course, we're interested in Arabidopsis, but vernalization is very important not only for many natural plant species, but also has been hugely important for agriculture. It's basically been bred in most of the major crops and significantly extended geographical range of many of the cereals and the brassicas. We know a lot about the genetics of vernalization requirement in many crops, but not very much about how those plants are actually perceiving the cold. In Arabidopsis in '99, two labs, Rick Amsino's lab in Madison and Candy Sheldon in Liz Dennis's lab, cloned this gene FLC. And this is really the central gene in vernalization. Now, the flowering field gene nomenclature is fairly horrible. There's lots of genes beginning with F or V or what have you. I'm going to try quite hard not to get into many of those today. I'll sort of describe them. But there will be one gene that I will refer to, and this is the one gene name you have to remember. And this is FLC, flowering locus C. It's this repressor of flowering that I showed schematically at the beginning. It encodes a MADS-box protein that basically transcriptionally represses a whole suite of genes that are required for the meristome to switch to a floral fate. And how vernalization, how cold accelerates flowering is to slowly but surely suppress the expression of this repressor. You can see that in this northern here. And then after the expression is suppressed, it's then stably maintained at that low level through the rest of development. And it was at about this point we entered the game. What we wanted to understand was, well, how does a plant know if it's had 1 or 2 or 3 weeks of cold? Many cold processes, many temperature processes are much faster than that. Why is this so slow? And also, when it's at this low level, how is it then stably maintained at that low level through the rest of the life cycle? Which is, of course, important because often flowering occurs many months after the plant has experienced the cold. So we came at this with a classic forward genetics approach, just finding mutants in Arabidopsis that were defective in vernalization. Started with a vernalization responsive genotype, looked for mutants. Most of the plants would flower early after vernalization, and the mutants we wanted would be then flowering late. Initially, this was all done based on flowering time, and then once we knew that the target was this FLC gene, we did it based on FLC expression. And we've tried to go over and over and over and saturate for mutants that were defective in vernalization. Now, a whole suite of different genes and proteins came out of those screens. I'm not going to try and cover everything today, but the ones I will describe are a protein which is actually a homologue of a now well-known chromatin protein called SUSY12. In Arabidopsis speak, it's Vern2, but that's a horrible name, especially in Davis, in the confusion with the wheat field. We also found, I'll tell you about that, we also found some of these PhD or plant homeodomain proteins, and I'll tell you about those. We've also found DNA binding protein and a heterochromatin protein homolog, but I won't have time to get into that. Now, we cloned this first one, this SUSY12 homolog, many years ago now, but it took us into this field of polycomb regulation because this is the schematic representation of the Arabidopsis protein and then the Drosophila protein underneath. At the time, there wasn't much known about SUSY12, but fairly soon after we cloned the gene, it was clear that this was part of a small complex with two other proteins, and these are the other Drosophila names, that formed a polycomb repressive complex 2, or PRC2, and the activity of this three-protein complex was that it modified histone tails, specifically histone 3, lysine 27, and it gave trimethylation. Now, at the time, this was, well, another of those modifications, but this is really a modification that is very characteristic of polycomb silencing. Now, polycomb proteins were at first characterized in Drosophila as regulators that were important for regulating a body plan. They would function after other regulators and fix expression states of genes. And they were important because if you lost them, you would then end up getting the body parts in the wrong place of a fly. So this idea that they came along after other regulators and provided a memory was an attractive model for us, in that they perhaps were then providing this memory of this cold that the plants had had. So this was the initial idea of how a polycomb type mechanism might play an important role in vernalization. Several years later, then we kept going with the mutants, kept going with the cloning, kept going with the characterization. So, a lot of this is published, so I'm whizzing through the schematic, not too much detail overview here. We got two mutants, which I'm going to, the sort of published names were, well, Vin3 was the name that Ricamacino gave this mutant. We had called it Vern7. We got Vern5. These two mutants, which came out separately from our Vern screen, defined a little gene family in Arabidopsis, which I've shown schematically here. There were 4 intact genes, each with a PhD finger, a fibronectin 3 domain, and then a very conserved C-terminus. And there was a sort of little pseudogene runt which sat just alongside this one here. Now, as I say, these names have got fairly horrible, and because they're PhD proteins and because that's the probably important feature of them, I'm going to call them PhD protein. Okay, so one or two may find that sort of disconcerting because I've changed the gene names, but I think it'll make it easier to follow as I go through. So we knew both of these were important for vernalization. We didn't know how they fitted together with each other or how they fitted with our PRC2. But one thing was quite interesting was when we looked at the expression pattern of these genes. One of them, this PhD3, was cold inducible with a timing that was rather familiar to us in terms of it took a long time. It sort of mimicked the vernalization response in that the gene wasn't expressed without cold, non-vernalized, but you could see 2 weeks, 4 weeks, 8 weeks, the expression then increases. So that was interesting in terms of an association with this cold-induced silencing process. The second one, which we knew is important, was constitutively expressed. But what became clear quite quickly was this conserved C-terminus was very important for forming homo and heterodimers between this little gene family, or the protein family. Question was, though, still, how did this fit in with the polycomb, the PRC2? Well, that became clearer when we did an affinity purification of one of these, this PhD5 protein, and pulled out, so affinity tag, pull it out, put it in the mass spec. What do we get? Well, we got this list of proteins here. So these are the Arabidopsis names, and here are the Drosophila names. And if I show you schematically what they are, it's the 3 proteins of the PRC2 complex with an often associated complex, protein MSI1, and 3 of our little PhD family, 2 that had come out of the genetic screen and 1 that hadn't. Now, when we look in the Drosophila literature, there are actually modified PRC2 complexes that are important in Drosophila gene silencing, and they look, in our view, somewhat similar. So they've got the same 4 proteins here as these, just the names are different. And they also have a protein with PhD fingers. It's just that this one, called PCL in Drosophila and PHF1 in humans, has 2 PhD fingers instead of 1 each under each of these proteins. So we see them as functionally equivalent, but not homologous. And in fact, this complex, the work in Drosophila has shown, what the PhD protein does is turn up the activity of the methyltransferase of the PRC2 complex. So it's sort of like a superactive PRC2. And that's how we view this complex here. So how are all of these working on our target, FLC? Well, when we then look at how these different proteins are associated during the different phases of vernalization, we do this with chromatin IP. We look at the various parts of the vernalization process, and I'm going to show you schematically here where all of the proteins lie and the various modifications on the FLC gene. So let me just introduce you to that here. It has 7 exons, and this very large first intron will become important. This is the transcription start. The first surprise was before cold, our polycomb PRC2 complex was already associated with the gene. Now that was a surprise because in the literature, polycomb was sort of thought to be recruited as part of the silencing, but it was already associated in the state when it's normally expressed. What happened during the cold was that the PHD3 protein was, of course, activated expression-wise, and it formed this complex that I showed you in the previous slide, but it was associated at just one place in the gene, in this target, in the beginning of this large first intron. And that's all that happens during the cold. 4, 5, 6 weeks of cold. Then what happens when you bring the plants out? You lose expression of this cold-induced protein, and the other ones associate over the whole locus, these 2 PhD finger proteins. Remember, I said to you that what we see these PhD finger proteins is doing is superactivating the activity of the histone methyltransferase. So now what you see is a very high level of this histone modification, which is associated with then silencing of the gene. Now, we're actively working on putting these various other components that I said that came out of the genetic screen into this picture, but I'm not going to go into that today. But just to reinforce the difference between the sort of canonical, if you look in a textbook at polycomb silencing, you'll see that polycomb proteins are recruited to loci as they're silenced. That was the picture in the textbooks, that was the picture out of Drosophila. But in fact, the recent literature in the mammalian field, especially with the embryonic stem cells, and there's masses of papers every month that come out now on polycomb silencing in the ES cell system, looks more like the Arabidopsis system in that even in the on state, the core PRC2 is already there. There's something then changes during differentiation, either addition of accessory proteins or modification of the core PRC2, which leads to silencing and differentiation. And of course, for us, we would say this looks more like the situation in our vernalization. However, it's now looking like quite a conserved mechanism. We've got PhD proteins, we've got PRC2, we've got the same modification as polycomb silencing in all of the organisms. Yet vernalization has actually got some interesting features that make it quite attractive to us and rather basic questions about epigenetic silencing. For a start of, what's the basis of the quantitativeness of this whole mechanism? As I recapitulate here the northern I showed you. What's the dynamics in terms of what I'm calling nucleation, is this formation of this PhD PRC2 just in one place in the intron. And then once it comes back out into the warm, the sort of the spreading, if you like, of these PhD proteins over the whole locus, how does that happen? What's the timing of that? And also this real conundrum in the field of how are histone modifications copied? How is something that doesn't involve DNA methylation mitotically stable? We still really don't understand that mechanism. Now, what I'm going to talk about today is chiefly this, is some work we've been looking at in terms of the quantitative response. So we went in and tried to be more quantitative. So we went in with chromatin immunoprecipitation and looked now much more accurately across the whole gene at lots of different time points, looking at this modification, this characteristic K27 trimethylation. And what you can see here, and it looks sort of such a simple diagram, but this is so difficult to do, to get the chromatin IP reproducible enough to then look at a time series is really quite tricky. Very, very excellent postdoc Jia Song worked this up. And you're looking at here the profile of K27 in the blue. This is in plants that have had no cold. In red, that's had 2 weeks cold. And in yellow, that's had 4 weeks cold. And what you're seeing now is this increase of K27 just at this region where these PhD proteins are binding during the cold. And you see that peak increasing the longer the plants are in the cold. So we sort of discovered this as the volcano was going up last April. So I see this as this, here's our volcano peak erupting slowly, slowly. The more cold you have, the higher the peak. Then what happens when the plants come back into the warm? The same color coding. Our volcano then sort of flows down the sides, coats the gene. But again, that quantitativeness is maintained so that you have more K27 if you've had 4 weeks cold than if you've had 2 weeks cold. And it's these very high levels across the body of the gene that keep it then epigenetically silent for the rest of the life cycle of the plant. Now, at this point, you say, well, okay, so it's quantitative, fine, but, it really still doesn't tell you about the properties of the mechanism that we're working with here. And at this point, I was lucky enough to be able to bid for, I was telling some people today, for a new funding stream from Europe, called the European Research Council. But we had to do step change science, blue skies research. You couldn't do anything that you'd done before. So I was like, okay, how can I make what I've been doing? So you've got quite good at this if you want to work on something for 20 years. How can I make what I'm doing sound really new and interesting and step change science? So I actually was very lucky enough to start working with a project leader who's been a new hire at the John Innes, who was actually into the computational biology department. He was a theoretical physicist who was an expert on spatial and temporal modeling. And we had lots of conversations about, we have temporal and spatial dynamics of FLC. Could he help me with understanding the mechanism behind all this? And inspired by this paper, which is now relatively old, it was 2007, we decided we would have a go, put a bid in to actually do quantitative modeling of vernalization. And believe it or not, they funded us. So the next little bit of the talk was a very interesting adventure for both of us into quantitative modeling of Arabidopsis. Now, what did this paper say? Well, basically, it was all about fission yeast. And in fission yeast, you have different mating types, and they switch stochastically from one type to the other. There's quite a lot of genetics that's associated with histone modifications. In essence, you're either covered with, let's say, an M modification, methylation, but a silent modification, or an active modification. In their case, it was acetylation. It doesn't actually matter. But you have bistable states, and you flip from one to the other. And in between those, you're actually mitotically stable. And what they tried to do was to model it and suggested that if you're an M modification on your, this is a nucleosome here, you're more likely to recruit complexes which will give you more of that same modification. So in our M case, you'll give histone, you'll attract histone methyltransferases. You'll also attract the deacetylases in this case to get rid of the competing active mark. But it's a positive feedback loop. So if you have your M, you're going to recruit more complexes and create more M's, okay? And so what they did was then model it based on dynamics. So these turnovers were happening on a timescale of minutes, on, off, on, off. The whole thing is a dynamic equilibrium between M marks and A marks, which are actually quite stable, but just occasionally flip from one to the other. And this model required actually cooperativity. It needed to have 3 steps. It needed to go from M to U to A. I don't even really understand all the physics, but Martin does, so that's fine. But what I did understand, well, what you needed to have was long-range interactions. So that if this M here attracted a histone methyltransferase, let's look at this one, that histone methyltransferase could equally methylate this histone down here as the one next door. So there was no near neighbor preference. Okay, so can we put all of this into Arabidopsis speak? So what you're looking at here is 9 fake cells, could be from anywhere in the plant, where we've come at the end of the cold and we're looking at K27 trimethylation. So you see this peak in the intron. And what we're going to model now is our M modification. In our case, it's K27 trimethylation. But this will work for any silent or active mark, which is a bistable state. And there's a time, this noise in there where the histone modifications are turned over in the matter of minutes, nucleosomes are turned over on the sort of timescale of hours, and then of course you have DNA replication. And every time then the histones are diluted, you have to recreate whatever that mark is. Can you model that? In fact, what we found, I'll run it a couple of times, is suddenly what you find is there's a kind of switch from an active state, which is when the M is very low, you build up this peak. And at a certain point, that peak will flip the switch into now fully M over the whole locus, which is then actually stable through the rest of development. Let me just run it again. It also predicted timing of when this K27 trimethylation, the dynamics of accumulation. It also said that not all cells would flip, and when they don't flip, they then decay and you stay active. So what it also showed us, and let me just recapitulate this schematically. So what we're saying is that in our nucleation region, this is FLC, here's our K27. Remember, from the data I showed you, which is what we used to parameterize the model, we have increasing amounts of K27. So see it as a patch. In a sea of A, you're now having an increasing patch of M, which increases as the weeks go by. And suddenly, that will increase the probability of flipping the switch now into a spread state where you have very high M across the whole locus, which is now epigenetically stable. And because this increases with cold, and this then increases with cold, the quantitativeness comes about from a changing proportion of cells that are flipped into that stable M state. So actually the quantitativeness is not per cell, it's the number of cells that are flipped. Now this is actually, now when you think about it, you think, oh gosh, it's kind of obvious when you think about it, but you know, when you're grinding plants up and you look at quantitative changes, you're thinking quantitative changes per cell all the time. But I think this is coming out as actually quite a widespread phenomenon where quantitativeness is often a population-based thing and not per cell-based thing. Secondly, actually, when you think about the number of epigenetic mechanisms around, that there are very stable epigenetic mechanisms. And so to be stable, you have to obviously, have some kind of mechanism to recreate yourself, in which case, how can you switch? So there has to be noise, there has to be dynamics, as I've described, to be able to switch from one state to the other. So we think this might be actually quite an interesting paradigm, let's say, for epigenetics on a more wider level. Now, what I've been telling you so far, go back, remember, we're interested in this cold-induced silencing. So far, all I've told you about in terms of the cold bit is the accumulation of the PhD protein, this Fin3 protein. Now, is that the only cold step? When we looked at the dynamics of the expression, and we've got a schematic here, so here's our autumn, warm, we've flipped now into our period of cold, and if you look at FLC expression, this is the RNA, spliced RNA, this is what sort of I showed you on the northern, slowly but surely goes down in the cold. What was quite interesting was when we then started to try and assay transcription, and so we can't, it's quite a low expression, so the way we've done this is actually look at unspliced transcript, I call nascent sense here. That had a slightly different profile. So that's suggesting the RNA turnover has also got interesting differences in the warm and the cold, which is actually different from the transcription rate. When you then look at the accumulation of this PhD protein, well, that sort of fits maybe with this going up and this coming down. So yes, maybe that's the only cold step. However, if you look at the silencing of our gene in now a mutant that doesn't make this PhD protein, it's exactly the same. Saying there must be some other earlier cold steps involved in the initial shutdown of the transcription of this target FLC. So that's brought us into the whole world of non-coding RNAs, which we've been doing for several years now. It's very confusing because, as I'll get into in a minute, there's two parallel pathways. This one I referred to as development in my second slide. We started genetically quite separate. All the mutants were separate, and they've both come down to the same target. Turns out chromatin regulation of the same target, and actually regulation via a non-coding RNA chromatin. So I apologize that when you look at the papers, you think, well, she's talking about all these different modifications. There are actually different pathways funneling into similar mechanisms. But let me just talk to you about the cold one here. Just remind you, here's our schematic FLC again. Here's our large first intron. This is what the sense transcript would look like when it's spliced. This is the nucleation site. Just little other features for those who are interested, but not terribly important right now for this talk. But what was important was, by analysis of some of these, took us into looking at these antisense transcripts of FLC. Now, what's interesting is that the 5' end of the antisense transcript is exactly in alignment with the end of the sense transcript. So the end of one is the beginning of the other at one end, and that's also true at the other end. The 3' end of the antisense is the beginning of the sense. In fact, the antisense just completely encompasses the whole of the sense transcript. The other thing that's unusual, and of course now there's a lot of literature about non-coding transcripts, but not many are spliced, and there's alternative splicing. That's what these little things are here. And also alternatively polyadenylated and capped. So they look actually very much like a sense transcript, except of course they're stuffed full of nonsense codons, and so they're turned over very rapidly. These don't accumulate to very high levels at all. But the interesting thing was when we then looked at the accumulation of these after a short period of cold. So remember, we're looking for something early in the cold process that is going to be causing transcriptional repression of our targets and as a sort of early prelude to the polycomb silencing. So what we find is upregulation of all sorts of different forms of our antisense transcript, an unspliced form, which is what this is supposed to represent, very high levels of this proximal polyadenylated site, and actually very high levels of this one here, which actually is alternatively spliced relative to the one I showed you before, and I'll get back to that in a minute. This is the northern just to show roughly the level of increase, but it's not very abundant. It's a bit misleading, this one. Now, we had known that these weren't absolutely crucial because you can make a version of FLC lacking this 3' end. In fact, the Australian group had done this, and you did get vernalization, so they're not absolutely required. But would they be sufficient? So we did that, answered that question by generating, oh, I forgot my joke slide. I always do this. Sorry, let me back up a bit. As we were doing this, the mammalian field, the polycomb field, got very excited by non-coding RNAs to the homeobox genes. And the first non-coding RNA was called hot air for Hox-associated RNA. So my postdoc thought it would be really hilarious to call this thing that we were working on, given it was cold-induced, cool air. So we have cool air transcripts. And, I thought, oh, I should have to live with this for years. But actually, it's very interesting. A name, people remember a name, so you can go around and, so we're the cool air kids. Anyway, so that's the joke. So anyway, we stuck cool air on to a GFP sensor, so we've got basically the 3' end of FLC, but the antisense promoter, versus just a control one that's not going to be affected by cold. And I hope you can see from this that what you get by addition of these cool air sequences is transient silencing by cold. And this was associated with antisense transcripts, cold-induced antisense transcripts. Now, we're still working away to figure out, are any of is it still involved in FLC? How is it involved in FLC? Initially, when we wrote this, we thought, well, a nice model might be transcriptional interference. If you upregulate transcription of the antisense, you're going to interfere with sense transcription. From quite detailed analysis of a custom tiling array we made, which tiled a single nucleotide resolution across both strands, it doesn't really look like that. You might say, well, we're making double-stranded RNA, but certainly in any of the RNAi mutants, vernalization is just fine. So we're looking at the model and trying to figure out what's happening. Of course, I think there's going to be a lot of redundancy here, which of course always complicates trying to figure out what's going on. But some clues have come from this parallel pathway that I worked on, that I referred to. Now, I'm only going to show this schematically, so I'm happy to go into more detail for anyone interested afterwards, but just keep it not too complicated. What I've told you so far, okay, just to recap, prolonged cold somehow transcriptionally silences the gene, and then you get accumulation of the PHD protein, nucleation of the K27 trimethylation in this region of the intron, associated with, and I've just put this purple bar for increased expression of the antisense. And then that is switched into this epigenetically silent state. So that's what we've done so far. The question is, what's up here, okay? And that's this other pathway. Now, as I said, we had started from genetics quite independently. That's taken us to the same target, to chromatin, but now actually a different modification, H3K4 dimethylation, which is associated with active transcription. But if you look, what I want you to notice is the transcripts, the antisense transcripts. So we have different states. Again, you could think of them as bistable states, high or low expression. And that actually affects flowering time in the plants without any cold. This is the famous autonomous pathway, or frigida. You have changed processing of the antisense transcript. Now, let's just focus on this one here, low expression. With low expression, you have low levels of K4 dimethylation, so no green blobs. Because you have low levels of K4 dimethylation, you have low transcription. If you have low transcription, you then favor this splice, this little intron splicing here, which brings in an exon, which brings a polydynylation to the proximal site, which somehow triggers recruitment of a histone demethylase, which keeps K4 dimethylation levels low, which keeps the transcription rate low. So you have a little positive feedback loop that keeps the low expression state. And in the absence or the perturbation of any of these splicing, 3' processing, RNA binding, you trip into this high expression state, which is also actually quite stable. Now, why I pointed out that changed splicing pattern in the cold is because here we have this alternative splicing associated with this. It's called the kinetic coupling of transcription and processing. It's now becoming fairly well established in yeast that this is the elongation rate is intimately connected with the splice site choice of many transcripts. And I think this is what's happening. Somehow in the cold, we're flipping from a high expression state through a dynamic positive feedback loop into a low expression state involving somehow this antisense transcript still. So we're still hand-waving at this point. You know, we don't quite know all the details yet. But this then causes the silencing. It's involving the antisense transcripts. And then, of course, you have a second RNA phase, or second cold, I should say, phase, causing the PHD accumulation and the beginning of the silencing. But it's not even as simple as that, if you think that was horrible enough. When you actually look, this antisense transcript does immunoprecipitate with the PRC2 complex, as it does actually for many mammalian systems. So there's this kind of complex interweaving of all these regulatory things together, and somehow we're going to have to unpick all of this to really understand this whole process. But kind of this is where we're at the moment. Now I'm just going to end on a slightly easier note, you may be happy to hear, but with, again, a different trajectory from the lab, which has taken us back straight into our chromatin mechanism, but is providing tools that I think are going to be quite useful for unpicking some of this complexity here. And that's natural variation in this whole process. So what I've talked to you about so far is, you know, vernalization in 4 weeks, 6 weeks. We put them in the cold room, the sort of lab winter. But if you collect Arabidopsis from different parts of the world, you need different weeks of cold to satisfy the vernalization requirement. And I've just shown 4 here. This one actually originated supposedly from the Botanic Gardens in Edinburgh. Flowers very late indeed, but if you give it 4 weeks, flowering's accelerated almost to saturation. Some of these other ones, which happily, turns out, came from Sweden, but we didn't select them on that basis. We selected them that they had some interesting vernalization responses. And if you look at this orange one, you give it 4 weeks, hasn't done anything to accelerating flowering. So very different dynamics. This one I'll come back to. This is called Love 1. This is from almost at the Arctic Circle up in northern Sweden. So we've been doing quite a lot of work, because if you think, well, maybe we understand vernalization enough to actually be able to explain all this natural variation. So we went in and had a look at what was happening. And again, I'll just show most of this schematically. In our Edinburgh type, or actually the more familiar to you Columbia type with a frigida in it, you have very high expression in autumn. And then during the cold, remember 4 weeks was almost enough to fully saturate, FLC expression comes down. 4 weeks is enough to then epigenetically silence FLC, that allele. However, if you do the same experiment from this northern Swedish type, 4 weeks isn't enough to epigenetically silence the allele. It reactivates. But it's not that there's anything wrong with the machinery for vernalization, because if you give it longer cold, in this case 12 weeks, I've shown schematically here, it silences perfectly well. So what that is saying is that something is accumulating during the cold, which is quantitatively contributing then to the epigenetic silencing post-cold. Of course, the beauty of the Arabidopsis is you can cross those and then map the determinants that are underlying that variation. And I whiz through this again. What was interesting, we did quite a few crosses, trying to pick, actually really trying hard to pick different ones, thinking we might end up working on different genes. And actually, for all of them, there were common QTLs, one of which was smack over our target, FLC again. And interestingly, none of them mapped to the trans factors, none mapped to the VIN3. You might have imagined the accumulation of this PhD protein might be different in different accessions. That doesn't seem to be the case. We've done it, and it also doesn't map there. So that suggested FLC itself was involved, and we've now gone on and cloned the alleles and put them into a common background. In fact, this was the very early background, which is an FLC null. Here's the allele which is saturated by 4 weeks, and this is now flowering after a 4-week vernalization. This one's flowered. Here's the allele from the northern Swedish one, and it hasn't. So that clone, which happens to be 10 kb in this particular transformation experiment, carries all the information for this quantitative variation in epigenetic silencing. The other really cool thing was then when we looked at the K27 trimethylation, so remember our volcano going up slowly but surely in the cold. This is the cold fry, which is basically what I showed before. 4 weeks, it pretty much saturates the K27 trimethylation at the peak, this nucleation peak here. This is in our responder that saturates at 4 weeks. However, if we do the same experiment in our northern Swedish one, here's what 2 weeks looks like. Here's what 4 weeks looks like. It's not as high as this. It takes 9 weeks to get to the same level, or fountainhead level, to then trip into our fully epigenetic silent state. So when we then look and say, well, where are the cis elements that are underlying this variation? So here's a schematic of the Columbia allele with the various bits that I've talked to you about, this nucleation region at the beginning of the intron, the antisense promoter. Here's the love allele, this northern Swedish one, and there are about 25 differences. The red shows the SNPs, and these little triangles shows various insertions. And the data what we've done so far is to make mix and match of these alleles to ask, so which bit of all of these polymorphism is conferring the different quantitative memory. And so here's our red is the late one, black is the earlier one, and we've done these various mix and match versions, which the postdoc thinks this is a really simple way. There are 5 forms. So if it's LLLLC, it's just the C at the end. Anyway, that's the sort of the idea of this now, which is the data slide. So we're getting near the end here. If you then look at transformants, and we have to do this on pool T2s to actually get a quantitative number with all this position effect problem you have in transformation experiments. But here's our rapidly silenced allele type. All C's, having had 4 weeks cold. If you look 10 days, 20 days, 30 days, then it's stable. Here's our unstable one after 4 weeks. You see the instability. And then these are the various mix and match versions. And in fact, if I just go to this again, where it's these set here that are conferring the instability of the expression. So this is as far as we've got, but it's interesting that this is the nucleation region. So how we interpret this are these polymorphisms somehow. Is this a cis element? Is this affecting the cooler RNA structure? Is this part of the nucleation process? We don't know yet. But actually, because we've got perturbation, these polymorphisms may actually help us unpick what actually nucleation means. So this is the sort of model of the currently sort of intersection yet again of how natural variation has taken us straight back into the chromatin silencing mechanism. But it is nice because I think these polymorphisms will help us dissect these dynamics, understand what the RNA is doing, because the secondary structure of these non-coding RNAs seemingly is very important, and also provide insights into the epigenetic stability. Very quickly, I just don't want to leave with the idea that this northern Swedish one is the only useful one, because if you look and scan now across all the Arabidopsis accessions, and we do that by taking a measure, okay, so you take expression at 30 days after transfer, T30, as a ratio to T10, and you can see if it's a high value, you're a very unstable accession. If you do that now in a whole range of accessions, you see this bewildering array of variation in vernalization response in Arabidopsis accessions. These are the ones we've done the QTLs for, these maps to FLC. Of course, now we're working with Magnus Nobel to do association analysis of what's determining all this variation. And a lot of it does appear to be independent cis element variation at FLC. And the other, of course, thing is that actually it takes us into now more generally adaptation. Here we have modulations, tweaks of epigenetic silencing that, in my view, is very important for perhaps adaptation to specific habitats. Is this what's happening? Is this what this massive variation is representing? And so we're focusing at the moment on Sweden. Here's the LOVE1 accession I was telling you about. Here's another couple. Now, when you look at this, we have, I think, 250 Swedish accessions at the moment we're doing the genome-wide association analysis with. You might look at this and say, well, okay, these are fairly close together. They're going to be adapted to rather similar things. However, when you then, and of course I can now happily go around to Sweden on various field trips, looking at all these various sites. So this is the northern site. This is right at the, as I say, is a World Heritage Site looking out over the Baltic. I mean, this is under snow 4 months of the year. And these things, we've monitored them, we're following. There's an ecologist up there monitoring what they're doing. This is basically, often you have slips of mudslides. So there's a very harsh habitat for this LOVE1 accession. But the two from the south, one turns out to be from a field, which is a very unusual habitat for Arabidopsis, a field that, because it's Magnus's uncle's field, we know a lot about. It's been not tilled for 50 years. This is the other one. It's actually from the Baltic coast, where there are millions and millions of Arabidopsis growing here, presumably salt tolerant, quite different leaves, very different root structure. And so these are the first 3 sites that actually now we have funding and say, this is the Royal We, this is Joy Bergelson and Magnus are doing field experiments to actually ask, is this adaptation, is this variation really adaptive to these specific habitats? So let me just leave it there. I just want to show you a picture of the various people in my lab at the moment. I haven't talked about everything. Pei-Jin Li has done the natural variation work. Jia Song, I just want to give credit, fantastic chromatin IP analysis that she's done. I think I've covered this. And then Andrew Angel is the postdoc doing the modeling, and Martin Howard is the PI who came as this theoretical physicist who we have very interesting conversations with, and our collaborators and our funders. And thanks for your attention.
Also the beginning of the intron, the first 400, where we have the lesions produced, the spring growth habits. So have you take a look at Apetala1, for example, if there are antisense transcripts there?
No, we haven't. We've only really focused on FLC, but Shumei Chen has looked at A-gammas, which is a very similar story. It's actually intron 2. And it looks like exactly the same story. And interestingly, the modeling suggests you have to be a certain size to be stable. A patch of N to be really stable needs to be a certain size. And so this may be why these introns are quite big. And actually, it's then interesting in some of the brassicas, I haven't gone into, we are doing some work on brassicas, some of those introns are quite small. So then the question is, are those the ones that are now not stable? So we haven't quite got to the point yet. But I think size matters in that case.
Yeah, all the springs have huge deletions.
Have deletions.
Huge deletions, not small deletions.
So will be nice to do.
I think it's really nice, Caroline, that the modelling recapitulates what you see in vivo. But do you think that there are tunable polycomb targets for, I guess, cell-autonomous processes? Because what you've described requires the cell to communicate.
Thank you for that question. What I meant to say, so when it says cell-autonomous, that just means that the switching doesn't need any information from its neighbours. But it's just the switching. It doesn't say the expression is cell-autonomous.
Right, I see.
Because you've got non-coding RNAs, you've got small RNAs that I didn't get into, you've got potentially even protein movement. I'm not absolutely sure even FLC is cell-autonomous. And certainly the downstream product, FT, is definitely not cell-autonomous. So, that is just a part of the switch, that you can do it without your neighbours. But it doesn't really then infer cell autonomy any further downstream in the mechanism.
So following up on that, is there any conceivable technique for actually demonstrating the differentiation between the actual cell nuclear states?
Yeah, well, so we
model is that there is a mosaic pattern.
Yeah. Can you see the salt and pepper pattern?
It seems to me proving that is really
Yeah, well, we've tried quite hard with translational fusions, bringing them out and asking, can you see some sort of dotty pattern? That's proven very hard because it's not a very abundant expressed gene. It's also, remember I pointed out to you that the RNA decayed with a slightly different profile to the transcription. So you've got all of this background RNA which really complicates those experiments. So what we're trying to do at the moment is tag the intron with one of these tags where you can direct GFP to the RNA, now to the intronic version, so that we can look at more directionally at transcription. But that's going to take a little while to just get high enough expression to be able to visualize it, because we want to put it under its own promoter. The whole point really is to sort of look at the native expression. So I hope the reviewers don't ask us that, but yeah, we've been battling with that one.
that's connected with another question that came up in my thinking about what you're talking about. And do you have any evidence about the relationship between the two alleles in the diploid cell?
As far as we can tell from all the transformation experiments, and this is where you put, say, a LOVE1 allele in a different background, it's all working in cis, that something isn't floating over and affecting the other one. Because they do, in the same plant, you can see the stable one silence after 4 weeks and the unstable one reactivate. But we haven't absolutely shown that's happening in the same cell. I think we perhaps need to start to do some hybrids and further monitor, the different things. We just haven't done that yet.
So if I understood correctly, the methylation doesn't spread until the plant returns to the worm.
Yeah.
But is that included in your model, or how?
It is, in that and I think that's interesting. Why? Well, if you leave it for 12 weeks, it slightly leaks a little bit, and because there's a lot more quantitative data than I actually showed. But it is really remarkable. Remember I said it happens within 6 days, so I think I've got one slide where we've then done 1 day, 2 days, 3 days, 4 days, 5 days, and you can see the PhD protein spreading across. I mean, I say spreading, actually, it's how we described it initially. I think it's the dynamics that the pool of the PRC2 is now PhD PRC2, so that the recruitment now covers the whole locus. But why does it not do it? I think that's then the molecular question that we need to go back in. Is it VIN3, the cold PhD one that holds the complex together, that prevents the others spreading until that has decayed away? for example. But those are questions that come out of the modelling, which we can now go with molecular tools. Yeah, but this very high level of K27 requires this PhD PRCT because it's a superactive methyltransferase.
Question. So when you talk about the accessions from the different locations, how big is the sampling from each location? And are you looking at all that variation at a location?
Yeah.
Is it there's a higher frequency of plants with that?
Yes. No.
Faster?
Absolutely. I think the answer to that is no. And it's been a sort of an enthusiast who over the years has sort of gone back every once in a while, and every time they've seen a plant sort of harvested them, but not systematically. Until recently, but these 3 sites now, we're doing very systematic analysis. There's temperature sensors there. We've gone back over 2 years. They go back every month because, of course, there could be, you happen to go in June and you'll sample that particular variant, and a month later there'll be a different one. So, yeah, we just need to do a lot more systematic analysis, and that's now happening. Someone based in Sweden driving between the north and the south. But I think it's ecologists haven't done a lot of work on Arabidopsis. This is sort of, because actually it wasn't sort of the obvious thing to do much ecology on. But there's so much of a resource of an information in terms of all the other molecular, population, genetics, that I think actually it's an interesting time for ecological questions to start kind of being addressed in Arabidopsis. And the nice thing with Sweden, that's why I said fortunately we picked Swedish ones, because certainly the northern region is not, there's no people for sort of 500 miles, so these are very undisturbed habitats. So unlike Central Europe, where there's been very recent mixing, we might have things that are adapted. So.
No further questions.